I would like to introduce Dr. David Malkin, MD, as our speaker for today. So thank you everyone for dialing in today and attending our second ALSF Childhood Cancer Seminar Series of 2026. My name is Justin Ingram, and I'm the Research Program Director at ALSF. And as I mentioned, it's my great pleasure to introduce Dr. David Malkin, MD, as our speaker for today. Dr. Malkin received his medical degree from the University of Toronto and completed his clinical training in pediatrics and pediatric hematology oncology at the Hospital for Sick Children. He's currently a professor of pediatrics and medical biophysics at the University of Toronto, and he holds a CIBC Children's Foundation Chair in Child Health Research, is a staff oncologist, director of the Cancer Genetics Program, and a senior scientist in the Genetics and Genome Biology Program at the Hospital for Sick Children, where he co-leads the Sick Kids Precision Child Health Initiative. He co-directs the Sick Kids Cancer Sequencing Program and the Pan-Canadian Precision Oncology for Young People Initiative, both of which have established pipelines to incorporate NGS into clinical trials for Canadian children and young adults with hard-to-cure cancers. During his postdoctoral training, Dr. Malkin made a seminal discovery linking germline mutations in the TP53 tumor suppressor gene to the high lifetime risk of multiple cancers in the Li-Fraumeni syndrome cancer predisposition syndrome. Dr. Malkin's research focuses on genetic mechanisms of cancer susceptibility, particularly in the context of TP53 and LFS, exploring the application of genomics to surveillance and treatment approaches for individuals at genetic risk for cancer. Dr. Malkin has received numerous awards recognizing his clinical, research, and mentorship work, most recently being elected a Fellow of the Royal Society of Canada and the American Association for Cancer Research, and receiving a King Charles III Coronation Medal for his contributions to Canada. His extensive international collaborations and recognized leadership in pediatric cancer genetics and oncology have facilitated his research objectives, which are to develop a fundamental understanding of the molecular basis of LSF specifically and cancer predisposition in general, and to apply this knowledge to transform the clinical management of children with cancer. Dr. Malkin has a strong relationship with ALSF, where he's a co-principal investigator on an ALSF Crazy 8 project focused on finding novel treatments for kids with cancer predispositions. So on behalf of ALSF, I wanted to thank Dr. Malkin for his time today, and we'll pass it over to him for his presentation titled Li-Fraumeni Syndrome, New Frontiers in Early Cancer Detection and Interception. And as a quick programming note, we'll have a Q&A session after the presentation, and you may ask questions to Dr. Malkin using the chat feature. So with that, I'll pass it over to you, Dr. Malkin, and thank you for your time today.
Great. Thanks very much, Justin and Connor, and thanks for the invitation to talk with you and tell a little bit about the story of Li-Fraumeni syndrome, and looking forward to sort of questions and discussion after. So just my only disclosure here is co-chairing the BSc at the NCI. I won't be discussing anything related to that here. It's a bit of an outline. We'll give a bit of a background of the field of P53 and Li-Fraumeni syndrome. It'll be pretty high level, but just at least to bring everybody up to speed. And then sort of really three major areas of focus. One is on the genotype-phenotype relationships of Li-Fraumeni syndrome, discussing some of the findings of deep genome sequencing of tumors and the germline, and what we're learning from that and how it might be applied in a translational way. And then towards the end, just some taste of new approaches to prediction, early detection, and in particular, novel therapies. So really, I find this slide, and we actually used it in presentations of the Crazy 8 proposal at the ALSF meetings, is that in the context of cancer genetics or cancer predisposition generally, I sort of think of five main themes, and clearly diagnosis, being able to recognize a predisposition is critically important, for, of course, awareness and developing management strategies, developing biologically rational therapies. There are very, very few for most of the vast majority of predisposition syndromes, and where most of the efforts have been in place for the last number of years have been in early detection technologies or approaches and surveillance, and we'll talk about that in the context of LFS. And then the exciting area, of course, is now moving into cancer interception and prevention, and is it even, even the concept of being able to prevent childhood cancers generally is being something everybody's dreamed about, but not really any headway. And so we think that the model of predisposition gives us an opportunity to explore this in particular. So Li-Fraumeni syndrome is really the paradigm. It was described by Fred Lee and Joe Fraumeni initially in 1969. They epidemiologically did an incredible study of over 600 children with rhabdomyosarcoma in particular. And in 5 of these families identified a striking family history, which was consistent possibly with the genetic predisposition, though, of course, at that time, there were no real tools to explore that. Both Fred and Joe were extraordinary mentors of mine during my training and all through my career to this point. And Fred passed away a number of years ago, and very sadly, Joe Fraumeni passed away 2 days ago. He was 94, I think, and still very active in thinking about amazingly astute mind, even right up until the last month or so, where he would always ask me to send him papers and things and then fire questions back to me. So we definitely miss them, but really these 2 were icons in the field and developed the field of cancer epidemiology. So 1969, this cluster of families was identified, but it wasn't really till 10 years later that there were some clues that would be helpful. So in 1979, Arnie Levine and David Lane co-discovered this protein, which was actually 54 kilodaltons, that was presented in transformed cells, NIH323 and embryonic carcinoma cells that were transformed with simian virus 40, and the large D antigen pulls down this 53 or 54 kilodalton protein, which subsequently became, of course, P53. And at the time, it was really thought that this was an oncogene, an oncogenic protein, because it was being studied in these transformed cells. Over the subsequent 10 years, there were a few people who were exploring this interesting protein. But it really wasn't until the late 1980s, 1989, when 2 really seminal discoveries came along. There were many actually in that year, but these 2 I highlight. One, Alan Bernstein's lab. He was at the Mount Sinai Hospital in Toronto Research Institute, and he developed the first transgenic mouse with P53, mutant P53 allele being overexpressed. And these mice developed a variety of cancers, not typical of what you see in Li-Fraumeni syndrome, but of course it's a mouse, not a human, so you might expect different phenotype. And then around the same time, Bert Vogelstein at Hopkins and Janice Nigro and Susie Baker and the group there did a large-scale sequencing of a wide variety of cancers and identified that there were mutations primarily in the DNA binding domain of P53 across a variety of cancers. We now know that at least 50% of human cancers have mutations in P53, and probably about 80+% have alterations in P53 function. And then I was fortunate to be doing my postdoc with Steve Friend and Fred in Boston at the time, and we were interested in exploring what causes Li-Fraumeni syndrome. And it was those 2 studies that actually clued us in, because initially we are actually looking at RB. Don't ask me why. It's not clear to this day why I was looking at RB, because that's never actually been described in Li-Fraumeni syndrome. But it was the only tumor suppressor known. However, we then started doing sequencing in 5 families that were collected by Fred at the National Cancer Institute, classic pedigrees, and using gold Sanger sequencing. For those of you who are younger in the audience, you won't have a clue what you're looking at here, but these are the way we did sequencing back then. And you could sequence about 250 bases over 6 or 7 days. And we identified mutations, all clustered, it turned out, in the exon 7, 8 of P53, and then a southern blot was done of tumors demonstrating loss of heterozygosity in the tumors of these patients. And so that led us to really understand that Li-Fraumeni was a genetic disease, and in fact, cancer was a genetic disease, because here was really the first elemental discovery that demonstrated that a mutation in a single gene, when inherited, could cause multiple cancers. Prior to that, the only observation was retinoblastoma in retinoblastoma, and then very shortly after the WT1 Wilms tumor gene was identified in Wilms tumor, but they were fairly narrow phenotypes. p53 was subsequently described by David Lane as sort of the guardian of the genome, and this figure from Scott Lowe's group really is it's sort of old, but it really highlights this incredible complexity of functions that p53 is involved with, and to make it perhaps a little bit less complicated, this is just a nice simplified figure that exogenous events that induce DNA damage, usually double-stranded breaks in the DNA molecule caused by any number of external factors, induces p53, which is a transcription factor, to activate other transcription factors, which then induce a variety of downstream effects on trying to either kill or induce suicide, apoptosis, or DNA repair and growth cycle arrest. So just a slight segue, we've over the years then developed a program to study Li-Fraumeni syndrome at the Hospital for Sick Children, the Research Institute, with colleagues in other centers in Canada primarily, but also with large-scale collaborations around the world, which have been absolutely fantastic, and I'll talk to those shortly. And really, we sort of have 5 projects, for want of a better word. We've been exploring what the immunophenotyping of Li-Fraumeni patients is, what is different about tumors that are driven by mutation in p53 in the germline. We're exploring the concept of newborn screening, and there's actually another ALSF Crazy 8 project led by Lisa Diller and Sharon Plon, also exploring this question, and could we ultimately think of actually newborn screening for children, period, and children at risk. Also, we'll talk more about this, about developing vaccines for tumor interference and prevention, and then using a variety of models, including zebrafish and mouse models, to really explore other ways for early detection and novel therapeutics. So then what is it about p53 that makes life complicated in terms of studying it in the context of predisposition? And so this is a nice figure that was published a number of years ago, where this cartoon shows the phenotype, the diverse phenotypes in individuals who carry germline mutations in p53 compared to those who have somatic, where the mutation is somatic and there's no germline event. And you can see that, the tumor types are similar, but the frequencies are really quite different. And to highlight that, colon cancer is extremely rare in the context of Li-Fraumeni syndrome. It definitely does occur, and it's part of the surveillance protocol in adults, but it is rare, with only about 1.5% of patients developing colon cancer, whereas we know that somatic mutations in p53 are very common in colorectal carcinoma. On the other hand, soft tissue sarcomas are very common in Li-Fraumeni syndrome. They were basically the initial entry point for Ed Lee and Joe Fraumeni to describe this syndrome, with at least 15% of patients developing soft tissue sarcomas. But somatic mutations in soft tissue sarcomas are actually quite rare, except in the context of relapse or refractory or metastatic disease. And then when you look at the hematopoietic system, there's a relatively similar frequency of mutations in p53, whether they be germline or not. So an early mutation, a germline mutation, does confer a very different cancer risk than cancer type risk than does a somatic mutation, even though the sort of the range of variants across the p53 molecule are actually quite similar. In addition to that, you see a spectrum of cancers that are dependent, that are age dependent. And in a sense, they're more or less similar to the ages you would expect. We see breast cancer peaking in young adulthood. So while breast cancer is, of course, extraordinarily common in the female population, in the context of Li-Fraumeni syndrome, it tends to occur 10 to 20 years younger than in the general population. And this can be applied to all the other cancers as well. Osteosarcomas in younger children, not as many teenagers, mostly around 8, 9, 10, 11 years old. Adrenocortical carcinoma is very common in the very young, and brain tumors as well. And then you get this interesting bimodal distribution where you have a very high incidence of cancer in the first 5 to 6 years of life. It sort of it comes down a little bit during teens. There's a secondary peak primarily driven by breast cancer, and then it kind of tails off as one gets older. And there's some suggestion that if one survives, if you will, without a cancer before the age of about 60, the actual risk of developing cancer is relatively small compared to what it is in the earlier age groups. In addition to that, there's a wide heterogeneity. And so this is a family that was shared with me. It's a small pedigree of a much larger one from Josh Schiffman in Utah, who studied this as well and is a great colleague. And you can see this autosomal dominant pattern of parents, multiple cancers, early ages of onset, and about 40% of cancers do occur before 25 years of age, so very relevant to the pediatric space that we work in, and the very high lifetime risk. But this mutation, which is a very common hotspot mutation, arginine 248 tryptophan, this is a patient of mine who I've been following for a number of years, who developed 5 cancers. There's no family history, so this was a de novo mutation. But you can see this one individual has 5 malignancies. He's now actually 22 years old. He's at university and doing very well. But here, a malignant melanoma at age 11 is almost unheard of in the general population. And so there's this heterogeneity, which makes life even more complex. And in addition to that, when you start looking at the genotypes, this is a paper that we published a number of years ago with Lisa Diller and colleagues, Lisa Teot at the Dana-Farber, when we had demonstrated with them that the subtype of rhabdomyosarcoma that most commonly associates with germline mutations is the anaplastic, very aggressive subtype. But what's interesting here is when you look at all the patients that were captured in this particular study, it's kind of hard to see this, but all the mutations in P53 are unusual. They're splice site mutations, they're nonsense mutations, duplications, and so on. You don't see the more common missense mutations. And so there suggests that there may be some cell type specificity in terms of the types of alterations in P53 that are able to be sustained and lead to malignant transformation. And at the same time, this study published a number of years ago with colleagues at the DKFZ in Heidelberg, Germany, who studied brain tumors. They were looking at medulloblastoma, and we shared a lot of the germline data with them, and then did some of the sequencing on germline, and found that the sonic hedgehog subtype were all the patients who had a germline mutation were exclusively of sonic hedgehog subtype. You didn't see that in any of the other medulloblastoma subtypes. That still holds true, and in fact, patients with sonic hedgehog medulloblastoma are recommended to have germline testing, and in fact, their outcomes tend to be very poor. Those patients with germline mutations in P53 tend to have very poor outcomes. The diversity in phenotype is also notable here. So this is a study which has shown that depending on where the mutation is, you actually appear to have a variance in the age of onset of cancer. And so a mutation at codon 282 tends to be associated with a much earlier age of onset than one, for example, here at codon 245. The challenge with this sort of study, though, as you saw from the pedigrees, whether it was Fred and Joe's original ones or the one from Josh, is that even within a family, you can have quite a large range of onset of tumors, and it begs the question of how accurate this is. I realized as I'm speaking that I meant to put a slide in and I completely forgot, is there are now a handful of what we call hypomorphs, and these are variants which are associated with a late onset only cancers, or at least it would appear to be only, then they are probably founder mutations entirely. And the most recently published one from Sweden is at codon 181, an arginine to histone mutation, which seems to be associated with primarily with breast cancer and almost always late onset. There's only 1 or 2 children. And that then suggests that the surveillance strategies might be different depending on what the variant is. And the last piece here is just going into mice for a second. So one of the P3 mutant mice is a ortholog of the human 175 mutation, one of the most common dominant or gain of function mutations. And Cheng Chan at Rutgers University did this nice study, which basically shows that depending on the genomic background of the mouse, you actually have a range of age of onset, which is really quite interesting. And one can imagine extrapolating that then to the humans and understanding perhaps there are variations in the genome at the genomic level of individuals, which alters their phenotype. And so putting a lot of this information together, Christian Kratz and Pierre Reynaud, the late Pierre Reynaud, and I and others put together this concept of the spectrum, an LFS spectrum that really Christian has been really excited about. And it goes to this idea that if you have classic pedigree with classic dominant mutation, if you will, the pathogenic mutation, that's classic Li-Fraumeni syndrome. But then you get these more attenuated phenotypes where the phenotype doesn't quite fit what the classic definitions of Li-Fraumeni or the more current definitions of Li-Fraumeni. And so this adjusts the way that one manages these patients, and even one does cascade testing, surveillance, and so on. And so the concept of Li-Fraumeni being a spectrum of disease rather than a single entity is very important. So the idea was that, well, we can't treat, we can't prevent. And so some 20-something years ago, developed this concept that maybe at the best we could actually at least do some early detection, because we generally believe in all of us as oncologists, whether pediatric or adult, that early detection is associated with better outcomes as a rule. So we put together this protocol. This is the pediatric component of it. And it's been sort of dubbed the Toronto Protocol, not by us because we're Canadian and we tend not to do that sort of thing, but some of our US colleagues called it the Toronto Protocol, which is kind of cool. So we've used a variety of blood tests, urinary adrenocortical platform, whole body MRI, brain MRI, and then especially the physical exam. And I think a lot of folks, including ourselves, tend to rely a little bit too much, if you will, on these aspects. And I always guess go back to my old medical school days and learning from some of the best clinicians I could ever imagine is a physical exam can beat out almost any form of imaging and blood work if done well. And so we really, really spend a lot of time clarifying by history, by physical exam, anything that might be considered. And so with this in mind, we did this original study and a couple of interesting findings, which then leads to everything else I'm going to be talking about here, is that when you look at the patients that tumors that were detected by surveillance, there were a number of them, many of them, that were pre-malignant or low grade. There were definitely malignant tumors, but interestingly enough, almost all of them only required a surgical resection or relatively low doses or low aspects of chemotherapy for their management. And so about 2/3 of patients diagnosed by surveillance had benign pre-malignant low-grade tumors, whereas all those patients who presented clinically who were not undergoing surveillance had malignant disease at the time of diagnosis. And with this in mind, then we demonstrated that there was a very high likelihood of developing cancer in these patients, but you could pick them up early. And this is an example of a girl that I have been following since before she was born. She was diagnosed on prenatal testing and found to be a carrier, but carried to term. And at age 14, she had a whole body MRI, which, take my word for it, was considered normal. A year later, she came in for whole body MRI, and there was clear marrow infiltration. And 4 weeks before, we had seen her just for her regular blood work and so on, and everything was fine. She was asymptomatic. And in fact, on the day of this scan, in calling her, she was also essentially asymptomatic. But it turned out that she had a diagnosis of pre-B ALL, masked hypodiploid ALL. And at day 28, remission marrow showed that she still had 5% minimal residual disease. So picking up leukemia early, I don't know what that means because, in fact, she clearly had very highly resistant disease. And we made a decision at that time to actually take her directly to CAR T cell rather than transplant and more toxic therapy, genotoxic therapy, on the presumption that A, she was resistant to a lot of these drugs, and B, probably would be a very high risk of recurrence, relapse, and especially second malignancies. And she's now 200 plus stage. She's actually in university. And I saw her last week. She's 19 years old and transitioned to the adult program. There's Juliet and her mom. Lots of stuff. If you go just into social media, they're all over social media. And this study was led by Ron Rabinovitz, which led to a study which is now under review, international study looking at the outcome of patients with children with ALL. Who are treated with various forms of therapy, all who have germline mutations. And it actually suggests that immunotherapies seem to be a very good option for these patients. And so the original paper that Anita Villani, is a wonderful colleague, co-leads the cancer genetics program and the precision oncology program here with me, published this. And if there are any residents or fellow watching, Anita was a resident when she published the first paper in 2011, and she was a fellow when she published the second paper in 2016, and is being able to build her career around that. And we can see this very significant difference in overall survival. Yoshiko Nakano, who is a fellow with us now from Japan, is just completing an analysis, a long-term analysis. We're now looking at 20 years of follow-up, and we can see here that the surveillance is still holding true. This isn't an actually complete figure because we're still accumulating some of the longest term data, but we're pretty sure this is going to hold still. So back in 20 years ago now, it seems, Adam Schlein, who was a PhD student at the time and now has been on faculty here for a number of years. Many of you may know him through a lot of his work in the genomics of sarcomas in particular. But at the time, he was looking at copy number variation, and we were interested in whether there was sort of vertical transmission of DNA damaged DNA, which might account for this sort of accelerated age of onset in patients. And we believe that we were able to show that. And so Adam developed this sort of model of what was going on. And it was interesting because it was the journal wanted the model. We hadn't written this in, and then they said, oh, we have to put some sort of a figure of what we think is happening. And so we thought that patients are born, they have a germline mutation by definition. They probably also inherit or acquire a postmeiotic events or early postmeiotic events in these other modifier genes, which then leads to sort of various alterations of telomere attrition, chromosomal alterations, and accumulated genomic damage and accumulated copy number variation and structural variations, leading to this catastrophic event such as chromothripsis, perhaps, which is then irreversible, and that leads to malignant transformation. And the idea would be that surveillance comes in here. And so even though these events may not be able to be prevented, as long as you're able to jump in at this point before, then these patients have potentially a better outcome. So with this model in mind and everything we thought, Nick Light, who's an MD PhD student in the lab at the time and is now doing his residency in medical oncology, did a deep sequencing of a large, relatively speaking, cohort of tumors, a variety of different types from patients with a variety of p53 mutations using whole genome sequencing as the primary tool. And discovered sort of a few interesting things. So the first that I show here is looking at genomic signatures. Identified first that there's the aging or clock-like signature is found in virtually all patients, and we'll get back to that in a minute. But also, if you look at these two here, and actually there's another one over here, there's this kind of purple signature. And what that is actually a molecular scar of cisplatinum-associated insult. And Nick, of course, only had numbers. He had blinded himself, so he didn't really try to figure out, well, where did this come from? But it turns out that both these patients he was sequencing their second malignancy, and they both had previously received platinum for a prior malignancy. And this would have been years later, suggesting that this molecular scar is still present. What's actually very exciting is Adam has just, a paper has just accepted coming out in Nature now, where he's explored this not in the Li-Fraumeni context, but in children with sporadic cancers through our precision oncology cohort and data set, and really has identified that these molecular scars exist all over the place, whether they're platinum or anthracycline or others, and are probably playing a very significant role in the induction of second malignancies. Now, we've always assumed that to be the case. We all have plenty of preclinical evidence, and you treat various cells and things that this happens, but this is really the first evidence at a genomic level of what is going on and what alterations actually occur. The second thing is we were looking to see, is there a kind of a signature that is unique to patients with Li-Fraumeni syndrome that you could imagine detecting in the blood, and maybe would be a surrogate for all the surveillance, which is pretty intense, all the clinical surveillance tools. Could we develop a blood biomarker? And to do that, though, we would need to have a signature that is unique to Li-Fraumeni or to a germline p53 mutation carrier, regardless of the tumors that they might develop. So when Nick was doing the sequencing, these are tumors on each of these columns would be a different tumor that he sequenced. And the top are just some of the demographics. Here was the signatures that I showed you previously. And here are just a snapshot of some of the single nucleotide variants, the SNVs, in various different genes, which are sort of along this axis here. And this happens to be just a cluster of DNA repair genes. And you can kind of get the sense that every tumor has a different pattern of variants. There's no real two that look alike. There are some variants that seem to be recurrent, but not really that many. And this was kind of disappointing in the sense that we say, okay, well, there's no obvious signature that we could imagine creating some sort of biomarker for. And each of these tumors are different, and each of them express different mutations in these tumors. This then becomes an issue, as we'll see towards the end, when we start thinking of ways that we can actually target the tumors in Li-Fraumeni syndrome. Because if every patient's tumor is different, then you have to start to think about, rather than an off-the-shelf approach to therapy, it's more of a patient-specific approach to therapy. One thing that we did find in about 50% of patients is they all exhibit chromothripsis. Now, the concept of chromothripsis in osteosarcoma has been really pretty well established, but we were seeing it in a variety of other tumor types, like in this patient with an embryonal rhabdomyosarcoma, suggesting, in fact, that this is a more global event. And so perhaps looking at chromothripsis in the context of circulating tumor DNA or other analytes might be of value. And then the third part was looking at this concept of the aging signature or the clock-like signature. And so using some computational tools, Nick and company were able to sort of postulate then that the initial event is a germline mutation. You have loss of heterozygosity of p53 at some point. We'll see that in a couple of slides, a little bit more detail. But these events occur years before the actual transformative events in the tumors. So this sort of long period of time actually molecularly resembles the dormant, if you will, state that we see in the context of surveillance. And even in patients that we've done serial surveillance using whole body MRI, we have gone back and looked at MRIs of patients who do eventually get a true full-blown cancer, and find that in some of them we can actually see that there was a imaging abnormality months or years before that was just not really appreciated, if you will, radiologically as abnormal. As our radiologists have become more and more familiar and confident in the interpretation, and we do a lot of whole body MRI here, those misses, if you will, have become far less frequently. So our false negative rate on whole body MRI has almost plummeted to zero. We do get false positives, and then we follow them up accordingly. And in addition, these were 3 patients who coincidentally had in their tumors 3 different alterations, but all of them you can see are druggable. So in this patient had a BRAF V600E in the tumor. This one had hypermutation, eligible, and it was treated with immunotherapy. And then this patient had an F1 mutation, and unfortunately, she did not survive her myeloid sarcoma with her sixth malignancy and highly aggressive. So what is it then about this early loss of heterozygosity event, and can we learn something from it? What is actually happening here that is inducing these subsequent events in the patient? And so this is sort of just a blown up version of Nick's figure, and you start off with a mutant and then you get loss of heterozygosity here. We find that loss of heterozygosity is much more common, if you will, in the context of Li-Fraumeni syndrome than it is in the context of sporadic cancers, which is of some interest. But you can see here it's still thought to be really quite early. We also, just looking at this, looking for copy number loss, we see in the vast majority of the tumors, the copy number loss is a very early event. So in the last few minutes, I just want to shift gears to talk a little bit then about the whole concept of thinking of different ways to strategize surveillance. And working with Trevor Pugh, a brilliant computational genomicist in Toronto, who has a great interest in circulating DNA, circulating free DNA. The concept being that all cells shed DNA into the circulation, and these can be now detected with various technologies and use various technologies to identify different alterations, methylation pattern, genome sequencing, rearrangement, so on and so forth. So with Derek Wong, who's now at UPenn, we took the cohort of both adult and pediatric cancers, did targeted panel, shallow genome, and cell-free methylated DNA sequencing in a wide variety of patients who did or did not have cancer at the time. And this is just sort of the technologies again. And using these three primary modalities, we can actually look for a variety of sort of markers, if you will, that we use. And I'll show you that coming up now. So if we take, for example, this cohort of patients, so these would be a number of patients who had cancer at the time that their blood was drawn. Here are patients who did not have cancer at the time, or at least not clinically detectable cancer at the time. And they all have a germline mutation. About a third of them had a secondary p53 mutation that was detected in ctDNA. A few other tumor-specific alterations or tumor-associated alterations, and then sort of a group of patients where there's no single nucleotide variant that could be detected. This second group is particularly interesting. These patients don't have a clinical diagnosis of cancer, yet a small number of them, about 30% here, have alterations, either a second hit in p53 or alterations in other cancer-associated genes, suggesting that something is going on. There are cells that are demonstrating that they have alterations in other cancer genes. Are these ultimately going on to develop cancer? And in fact, they do. So a number of the patients who had these alterations months before they developed, months before, subsequently one had gone on to develop one malignancy or another. And this is sort of summarizing a lot of the data here. But basically what it shows is that for each individual patient, whatever stage of disease they may or may not have developed, they have different patterns of alterations. Some have shortened fragment size, which is DNA fragment size associated with cancer. Some have copy number alterations. There's a breast methylation score, and these patients had gone on to develop breast cancer, pan-cancer methylation score. But all of them have sort of a different pattern, suggesting that if we are developing a biomarker, we're going to need to develop one that actually is a multi-omic, if you will, approach, rather than one single entity. And that's a big difference from doing circulating tumor DNA as a marker in sporadic cancer, where you already know the molecular architecture of the cancer, so you know what you're looking for. Here, we don't know what we're looking for because every tumor appears to be different. There's now a clinical trial, which is a Canadian clinical trial, but is being expanded internationally, looking at 1,000 patients, both with Li-Fraumeni as well as hereditary breast ovarian cancer, Lynch syndrome, and neurofibromatosis, where blood is being collected every 3 to 4 months, and then patients can undergo their regular surveillance. And then on this arm, if the assay is done, if they find an alteration, then the patient is called back to start hunting down where the tumor is. And then the control arm, if you will, is the blood is being collected and will only be looked at retrospectively. And this is up till a couple of months ago. Accrual is going reasonably well. And there have been a few positives that have been detected. And in 5 of those 11 patients, they actually had a cancer detected on subsequent imaging. And the idea then is that the future state will be getting away from a lot of the intense screening that we currently have to deal with, and do a much more targeted precision approach to do this. Whether the cost will be, we think the cost will be reduced. At the moment, the costs are about $3,000 per sequencing, but those will probably drop to around $500 Canadian, which is, I don't know how, 600 US in the coming year or so. And then the last couple of minutes then is just a shift there, is that we know, and you've sort of learned a little here, that p53 variants do associate with different phenotypes, and they are also associated with a different grade or severity. And so work that is done by Émilie Montilier, is an amazing scientist in Grenoble, France, and published earlier this year, really demonstrates that there are associations of different p53 variants with different age of onset of cancer. But also, and very importantly, I guide you to the picture, or the figure rather, on the right-hand side of the screen, which shows us the immunophenotyping. And what it turns out to that is that the neoantigenic properties of p53 itself, of mutant p53, vary quite widely. And in fact, the immunogenicity of the most common germline mutations is actually relatively low compared to those of some of the more rare mutations. And why is this important? Well, if we're starting to think of developing a vaccine to target for these patients, then we need to understand how they'll respond to that. And to do that, we started off building this ALSF Crazy 8 team that you heard right at the beginning, that was introduced. Uri and I co-lead. Uri is studying replication repair deficiency. We're doing Li-Fraumeni syndrome, a wide international group collecting and samples, sequencing, and so on. And we're working on the immunogenic properties of all these mutations, of these tumors, of the sequences, to try to find identify targets that you could then develop into an RNA vaccine, which is being led by Drew Weissman at UPenn, and then exploring this first in the mouse model, and then hopefully ultimately in humans. And so the idea of this study is really, as I've just mentioned, we are looking for common neoantigens, and then HLA typing them, expressing which ones are using immunopeptidomics to see which ones are going to presume or predicted to mount a proper response, and then use the ones to develop the vaccine. For the RRD, this seems to be working well. The problem is, for p53, there are way too many neoantigens, and they are not clustering very effectively. And so we're starting to rethink how we strategize this in the context of Li-Fraumeni. Again, you're targeting the tumors when they're extraordinarily small, so we're sort of intercepting rather than necessarily preventing. And this really is just showing the various technologies that are used in this. So looking down the road, then, I hope I've given you sort of some taste of field. It's studying a pretty rare or uncommon disorder, but it leads to a much better understanding of this incredibly common cancer gene called p53. We're looking at other ways to predict age of onset and modifiers of genetic risk. What is it about those elderly or older patients who don't seem to develop cancer? Why not? And looking at epigenetic modifiers of the phenotype, which may be important. As I mentioned, a lot of new work going on in the CtDNA world. And also, what I didn't touch on here was work exploring the microbiome risk. And we're doing some of that here, as well as Suzanne McFarland at CHOP. And then, as I mentioned, the newborn screening thing. So with that, just like to thank, this is my lab at one of our Christmas parties at the house, and thank everybody here, and especially all the families that work with us, and of course, the ALSF, which is right there. So thanks very much. Happy to answer any questions.
Great. Thank you, Dr. Malkin. And I encourage people listening to please post questions in the chat for Dr. Malkin, and we can read them off. So the first question is involving the immunogenicity around TP53, so towards the end of your talk. Is there anything that you could potentially add to help prime an immune system to help work with a vaccine to essentially help make T53 a little bit more immunogenic while either administering the vaccine or before you would administer a vaccine to help in that regard?
Yeah, so one of the things that it's a great question. Thank you. So one of the things we've been discussing with on both teams, if you will, is using immune checkpoint inhibition and immune checkpoint inhibitors to see if we can help drive T cells into that. So enhancing, if you will, the immune response that you might expect from the vaccine itself. So that will be one of the things we definitely have to want to and have to explore. Thanks.
Yeah. So I guess another question is a little bit more broad in spectrum. So in terms of what types of cancers that are developed from LSF, is there generally a pattern with specific mutations in TP53 that drive different types of cancers, or is it just more where they spontaneously develop with a large kind of widespread mutation in TP53?
Yeah, so it's an interesting point. So back in, I don't know, I think it was in the late 90s, early 2000s, one of the groups in the UK had with pretty large cohort out of Manchester, had kind of made some correlations between specific classes of p53 mutation, not necessarily specific mutations, but specific domains and tumor types. So there was an association with certain types of mutations and brain tumors, certain type with adrenal tumors and breast cancer, osteosarcoma. That really hasn't panned out over the years. And the brain tumor is probably a good example because you can't just cluster brain tumors. There's multiple different types of brain tumors from very different cells of origin. So it would be a little unusual to expect just one organ to be associated. But there are some associations. So there's a founder mutation in Brazil, which is codon 337, arginine to histidine. It's the DNA, sorry, it's in the tetramerization domain of p53. That does have a higher prevalence associated with adrenocortical carcinoma. It is not entirely clear why that is. There may be exogenous or other events happening within the cells, but it is associated. They still get other tumors, but a very high prevalence of adrenal tumors. And then I think I mentioned the one of these more hypomorphic mutations are definitely appearing to be associated with different specific cancer phenotypes, one of them in particular with breast cancer. So I think as we get a better understanding and more families and more collection, we're going to see this a little bit, but some of them just seem to be associated with pretty much every type of cancer you can imagine. Yeah.
Thank you. Another question, are patients typically made aware of their specific TP53 mutation and does screening vary depending on that?
So yes, I mean, I can speak to our place, and I think most places. So whenever patients are referred for testing, whether they're babies or adults, the testing is done, and then through the genetic counseling programs here in our cancer genetics programs and similar programs elsewhere, those test results would be reported back. And then the decision about surveillance is done. This is one of the biggest areas of discussion, if you will. We've recently updated through the AACR guidelines for Li-Fraumeni syndrome for surveillance, as well as other syndromes. The guidelines based on experience over the last 10, 15 years in different centers, the availability of certain tests in different centers around the world, and also what things may or may not be valuable. So, for example, the ability to detect leukemia early just by doing CBCs every 3 months, most people would say doesn't make much difference, and most places don't do that blood test anymore. Whole body MRI, there are challenges with insurance funding and so on. In Canada, we're fortunate we don't have that issue, but I know in certain areas in the US and other parts of the world that is. In other parts of the world, doing a whole body MRI is basically the annual income for a family, like in Malaysia or places like that. So so you modify it according to the jurisdiction or the element, but where possible, and that's why I really highlighted the physical exam history because it's such an important piece of the surveillance. But yeah, I think the bottom line is the recommendation is everybody does receive that back because the consequences of not doing surveillance are, I think, proven to be pretty significant.
Another question is, have there been any ancestry enrichment studies done in germline variants?
Huh. Um, I'm actually not sure. That's a very good question. There are so when one looks at the spectrum of variants across different ancestries, I'm not answering the question quite the way it's worded, I suppose. For the most part, there does not appear to be a lot of variation from. There was one slide I put up there, sort of geographic diversity, which doesn't necessarily associate with ethnic diversity. There doesn't appear to be. There are a small number of variants. There's one codon 47 that appears to be hyperrepresented in African Americans, particularly from the African sub-Saharan region of the world. The codon 72, there's a variant of polymorphism that actually seems to be associated with latitude. So the higher more the Scandinavian countries, it flips more to an arginine, whereas in the lower Mediterranean, it's more proline, I think. So there are these kind of unusual things, but that's about as much as I can say. There are certain populations that everybody's now really exploring, and we actually are just working on a collaboration in India and in China, where very little sequencing has been done until to date. But my colleague in India at the Tata University or Medical Center in Mumbai has 350 families that they've now fully sequenced. And this is like an incredible rich resource to study. So they're really interested in working with us. So I think that might start addressing that question. It's a great question.
Another question is many of the multi-cancer early detection tests available commercially do not seem to be as sensitive for early stage disease. Would your multi-omics approach be expected to improve sensitivity for stage 1 cancers? And it seemed like the data that you showed there were higher stage cancers detected.
Yeah, so you're right. The slide that I showed, there were a lot of higher stage cancers, but in fact, I guide you to the paper where it's much more detailed, but we were picking up low stage, stage 1 cancers for sure. I don't off the top of my hand remember the numbers, but I think yes, in principle, the multi-omic approach should be better able to detect lower stage cancers, but that will clearly only come from the clinical trial. Yeah.
Okay. So, if there are any other questions for Dr. Malkin, you can put them in the chat now. But if not, I think we went through all the questions. So thank you so much. And we're right about at 1 o'clock. So we really appreciate the time today. And it was a fantastic presentation. So thank you so much.